Glycolysis is the first step of cell respiration where one glucose molecule is broken into two pyruvate molecules occurring in the cytoplasm.
In the process of glycolysis, two ATP molecules are hydrolyzed, and the first attaches to the phosphate group to one end of the glucose molecule, and the molecule then rearranges to fructose 6-phosphate. The second ATP attaches to a phosphate group to the other end of the fructose 6-phosphate molecule. The cone splits apart, and DHAP is converted to A3P, which starts the energy releasing stage. Each G3P gives one H atom and 2 electrons to NAD+, to become NADH. 1,3-biophosphoglycerate gives a phosphate group to ADP to form ATP. 3 PGs rearrange to become 2 PG, which is converted to PEP by the removal of a water molecule. PEP breaks down, releasing energy to transfer one phosphate group to ADP, forming ATP. The original glucose molecule has been broken down to 2 pyruvate molecules. The next stage of aerobic respiration then begins.
Pyruvate oxidation is the second stage in aerobic respiration. With the help of transport proteins, pyruvate molecules enter the matrix, where coenzyme A allows pyruvate to be oxidized and NADH is reduced to become NADH. The resulting product is acetyl-CoA. Krebs cycle occurs in the matrix of the mitochondria, and for every molecule of glucose, 2 Krebs cycles occur. Krebs cycle begins with 2 molecules of acetyl-CoA and 2 oxaloacetate molecules. Through a redox reaction, isocitrate is oxidized to become alpha-ketoglutarate, and an NAD+ ion gains electrons to form coenzyme NADH. Alpha-ketoglutarate forms succinyl-CoA by binding with coenzyme A and being oxidized by NAD+, which is reduced to form NADH as CO2 is removed. Succinyl-CoA becomes succinate as a phosphate from the matrix displaces coenzyme A and GDP becomes GTP. The phosphate group is transferred to ADP to create ATP. FADH oxidizes succinate to become FADH2. Succinate then becomes fumarate, which becomes malate when H2O is added. Malate undergoes a redox reaction with NAD+ to produce NADH and oxaloacetate. The cycle is then able to begin again.
ETC occurs in the inner mitochondria membrane. Proteins in the ETC are set up in order of increasing electronegativity. Electrons from NADH and FADH2 are passed from one component to the next through the membrane. When 2 electrons leave complex 4, they combine with oxygen and H ions to create water. This first process is done to create electrochemical gradient, which powers ATP synthesis. H ions are moved through the ATP synthesis, which is found in the inner membrane. The H ions flow into the matrix, which makes energy and creates a bond between ADP and PI, which creates ATP. At the end of the final stage, 36 ATP are produced in total.